Okay, here we're going to look at the process of gel electrophoresis. This takes the DNA fragments that are multiplied through the process of PCR, and I have another video on that, and separating them through literally a gel-like substance. And this is separating out those DNA fragments based on their relative size to one another. So where this may be used, we can typically see this being used potentially for paternity tests, forensic science, things like that, or how they determine who the father is. This process could be used for this purpose. What the basic setup looks like in the lab is a gel and a power source. Because we're using electricity to separate out the DNA, we need a power source. So here's the power source. DNA actually has a negative charge on its backbone. This allows us to have a positive charge located here, and that will actually pull the DNA down through the gel. We notice here the larger fragments kind of stay closer to where we initially put the well here for our DNA, and then the smaller fragments are actually traveling further through the gel material and getting closer to that positive charge. The gel electrophoresis, again, looking at our molecular size, which BP stands for base pairs. And we could see here the distance migrated. The greater the distance migrated, the smaller the molecular size. And we see that evident here. Here's our marker, which is just an area where we have known fragments of DNA. And we're seeing it separated out here. Keeping in mind those smaller base pairs, only 100 base pairs, are going further than those at 900 or 1,000. DNA is cut with restriction enzymes. So I have a video also on restriction enzymes that you may want to take a look at. And it's cutting the DNA at very specific sequences. This has many applications, such as forensics, paternity testing, medical diagnosis, evolutionary relationships, and many more. So comparing the cut-up DNA, how are the fragments compared? How are the fragments separated? They're actually separated based on their size, and their base pair length is specific how we're looking at size. Run through a gel substance, and you can see here it is literally gel-like. And it looks like initially, when we look at the straight lines, it looks like they all ran into the same spot. Well, actually, that is just the dye, and that is what's called the gel front. That is just showing you basically the furthest that the dye has moved through that gel. It gives you just an idea where the fragments are moving, but there will be little lines here, and we have ways of marking them and indicating where those bands may be located. So I mentioned that DNA had a negative charge, and it does. That negative charge comes from the oxygen, which is on our phosphorus backbone here. So nothing to do with the nucleotide bases. It has to do with the sugar backbone, and particularly the oxygens on the phosphate backbone. That's what's causing the negative charge. We notice here, this is a gel, we're on its side, and we have our positive charge here and our negative charge down here. So when we add our solution of different DNA fragments, we add them to the well, and we can see that here. And then the larger ones are not able to run as far. They're bigger and they kind of get caught up in this gel. The smaller ones are able to run further or closer to this positive charge. So the smaller they are, the further down the gels are going to run. And it's think of it swimming through gel. The bigger ones are going to have a harder time, and they're not going to move quite as far. How this looks, how we compare these fragments? Well, I mentioned this DNA ladder. Those smaller pieces moving further, larger pieces moving slower and lag behind. The DNA ladder is loaded into one of the gels. And what that ladder is there's an image of it here. It has known DNA base pair lengths. So we know what bands it's going to produce. This allows us to compare our unknown, or even our known segments to figure out how many base pairs long they are. The same process here, they fill the well up with the ladder, the smaller ones will run to the end, and the larger ones will run more towards the beginning here. And this is evident here. We have 1,000 base pairs, here's 500 base pairs, and here's 100 base pairs. When we're loading that actual gel, there are actually depressions in there. We're loading that mixture of DNA. Be sure to load these slowly so no sample is lost. It's a very small area. We want to make sure that we fill that with our sample. That shows it kind of on the side here, filling up that sample on the side. Be careful not to lose any. You'll see another well right here. Otherwise, that's going to cause a reduction in the amount of sample that can run through that gel. The basic process, they can be vertical or horizontal, those gels. We're taking our sample, here contains our DNA, we take our micropipetter, we're adding it to the wells, we add a charge to it, and then that's simply over time allowing it to separate out. Now it's important to not run it too long as far as time goes, because literally all of your samples will run out of the gel. You want to run it so they all separate, but so they don't leave the gel. You want to keep them in the gel so you're able to compare them. Running a gel, the DNA is cut with restriction enzymes, so that's what's giving the different fragments or different sizes. They stain the DNA with ethidium bromide, it binds to the DNA, and it's fluorescent under UV light. That's what causes it to glow. The ethidium bromide, remember, it binds with DNA, it can cause mutations, so that's why you always want to be wearing gloves with working with ethidium bromide. But what makes it really neat is that it causes DNA to glow under UV light. That's what causes these nice little bright bars or lines. This is indicating the size and the relative position of those DNA fragments. Again, the smaller ones will move further down, the smaller ones will move further, the larger ones will stay closer to the actual well. When using a lab, take extreme caution if using this, because it does mutate your DNA. We can take a picture of this with very special cameras and be able to tell us a little bit about the DNA. And if we're comparing samples, we can develop a DNA fingerprint, which is another video.